Good morning, everyone. Thank you for coming to this distinguished lecture. I just wanted to say a few words. Some of you may not know about the lecture series, and then introduce our speaker today. But the distinguished lecture series in causal discovery is sponsored by the Center for Causal Discovery. And the Center for Causal Discovery, as many of you may know, an NIH Big Data to Knowledge Center of Excellence that formally began in September of 2014, so it's about a year and a half old. And the major aims of the center are developing and disseminating algorithms and systems, software, that assist biomedical scientists in discovering causal knowledge from very large data sets. And another key aim is to train biomedical scientists and data scientists to use these methods effectively. So the center is sponsoring this lecture series in order to recognize leaders in the field of discovery and causal discovery, particularly in biomedicine, and to provide a forum for them to let us know about some of the cutting-edge developments that they're doing in discovery and biomedicine. Our speaker today, I'm delighted to be able to introduce Donna Pear, who is an associate professor in the Department of Biological Sciences and also in computer science at Columbia University. She obtained her bachelor's degree in mathematics in 1995, her master's in 99 in computer science, I believe. Donna, is that right? Yeah. And also a PhD in computer science in 2003, all from Hebrew University in Jerusalem. She worked with Amir Friedman on her PhD and worked with many other prominent people in the field. She did a postdoc in George Church's lab in the Department of Genetics at Harvard. So she has very deep knowledge in both computing and technology. And she's won a number of prizes: Overton Prize, Burroughs Wellcome, NIH Director's New Innovator Award, NSF Career Award, and others. And we're really delighted to have you here, Donna. Thanks so much.
Thanks for inviting me. Unfortunately, I'll talk very little about causation. But, when deciding what to talk about, you always stick a few things in that you feel like talking about for variation. And I guess maybe the most connected thing to causation is a lot of what I think about these days, or actually something that I've been thinking about for more than a decade, but is really coming to a point where we can actually finally really do it and regularly and possibly learn a lot, is the idea that you can learn networks directly from each individual cell. So our cells actually have a lot of natural stochasticity to them. Not all of them are the same thing. And if we measure their gene expression or their protein expression and look at their variability and treat them as samples, we can actually potentially learn networks, influences, interactions directly from these networks. So the basic idea is if we're measuring a protein and protein A activates protein B, then where the protein level is high, we can learn basically a function of protein A's influence of protein B directly from their single cells abundances and their variation. Asking the basic question, which we'll be asking throughout this entire talk: how does protein or later gene A influence protein B? And we're making a couple of big assumptions here. One, that molecular influences basically create statistical dependencies in the data we measured, which I really believe in, that we can treat each individual cell as sort of independent samples of the probability distribution of the co-occurrence of these variables that we're measuring, be it genes or proteins, which we can learn these dependencies from. So I'm actually going to raise a really old piece of work. The first time we attempted to do that at levels of single cells, or the first time that we realized that we could actually use single cells as samples from where we can learn. And this is joint work with Karen and Omer and with Gary Nolan's lab. And the basic idea was, this was actually a pretty unique data set at the time. It measured primary blood extracted from Stanford students. This blood was perturbed with different drugs that directly turn on and off particular variables that we measured. And we could measure up to 11 of these variables simultaneously in individual cells and collect thousands of cells. So it was pretty unprecedented just in terms of sample number, even forgetting the single cell resolution, of having 6,000 samples to try and learn from. And here, and that's one of the reasons I decided to bring this, we treated each perturbation, each drug perturbation, as an ideal intervention, and basically implemented Greg's algorithm into the code. I would like to say that this assumption is false. So while the ideal intervention assumption brought us a long way, in biology the ideal intervention is wrong. There's a lot of cycles there. Networks rewired. They're very dynamic beasts. There's a lot of redundancy and backup, and actually treating a drug perturbation as an ideal shutting down the variable with no other alterations to the network is quite wrong, but even this wrong assumption can get you pretty far. And in this case, it got us to pretty much the right answer. So in a data that was collected in an afternoon and an algorithm that ran 5 minutes on a computer, we could reconstruct 2 decades of painful and biochemical work about the interactions about these known molecules in T cell signaling. So these were primary CD4 T cells, and pretty much we got almost every edge right. We got a couple missed edges, particularly the feedback loops. So obviously, if we're trying to learn a Bayesian network, we are not going to get a feedback loop, so that wasn't very surprising. We got one edge going the wrong way, and actually that edge was the only edge where it was about almost close to the 50-50, and we actually didn't have any confidence on its direction. And particularly, we had sort of 2 edges that had some support. In the meantime, we have validation for both of them. But at the time, we decided that the influence of ERK on AKT was in 2005 quite unknown, and we decided to put our money where our mouth is and say we believe that there should be an influence of ERK on AKT. And so if we do another experiment and perturb ERK, we should see a change in AKT. And we used pKa as sort of like a negative sample, and that pKa shouldn't change much. So we wanted, since of course if you perturb a cell there'll be a change in everything, we wanted to show that the change in pKa is substantially less than the change in AKT when ERK is perturbed, and that in case was true. Now there's quite a bit of work showing that AKT and ERK have quite a bit of crosstalk between them. So that was the first piece of work, and that was done more than a decade ago. And yesterday, actually, Greg asked me, well, why didn't you do more of that? That was such a nice paper. And then silence. Oh, I forgot. And what we needed to succeed, we actually needed a lot of samples. This is downsampled to 420 samples, which was the largest microarray data set at the time, and it was pretty cruddy. And even when we took 420 samples but averaged them, so took 20 cells at a time and averaged them, it was even worse. So actually the actual single cell resolution rather than the averaging helped us, and so we needed both. So back to Greg's question, great paper, science breakthrough of the year, and then nothing. No breakthrough, no nothing, just silence. So basically that technology was painting with a wide brush. There are very few molecules that work for the FACS technology. It's very hard to get intercellular staining working. And I'll show you in a moment why it's painting in a wide brush. So, we get the stick figure, and we want to get accurate networks. We actually try to look at the data and understand more biology and go back beyond the stick figures of nodes and edges and try and say, how do these networks work? What are they doing? Exactly, what is the functional form of X's influences on Y? What can we learn about it? And beyond having some evidence that X influences Y in some way, we couldn't. So, we want to get to at least something a little bit better, maybe not perfect, but we wanted to get technologies that helped us better. And one of the reasons that this data was so poor, and one of the reasons that we couldn't really do much with this, as much as I banged my head against the wall, is the problem of spectral overlap. So this is the overlap of the best combination of 8 colors available. Remember, we were working with 11 colors, and when people out there say, hey, we have 18 color flow and we have 21 color flow, that's usually built for surface markers, and there's 2 pieces of cheating that these guys have going for them. One of them is surface markers are often expressed at fairly equal ranges, so everything is in the same order of magnitude expressed. And the other thing is they usually design their panels so if you want to get any given cell is only expressing maybe 4 to 5 of these variables. So while you are in your population of cells looking at 15, you are in any given cell only simultaneously measuring 4 or 5, and these panels are optimized for that. But when you're looking at signaling, which has orders of magnitude differences in their signaling, and each cell is expressing each of these signaling molecules, and you have the spectral overlap, then basically, let's say that this overlap right here is 1%, then, one molecule's noise or overlap will completely destroy the signal of another molecule. And that's where we were at, that the spectral overlap was killing any ability to do any quantitative signaling, and the signaling molecules had everything going wrong. We needed to measure all of them, and we needed to measure them quantitatively, and they were in different orders of magnitude. And here came in the CyTOF technology, which mostly was led by Gary. I did the computational undergrad. My job was to be the voodoo doll of the Nolan lab. They kept on sending me data, and I kept on telling them, no, this is more noise than signal, and trashed 2 months of a postdoc's work until one day I said, there's more signal than noise here, and we all celebrated. The idea behind this technology is, again, like flow cytometry. You take a cell, you freeze it, you poke holes into it, you stream these antibodies in, but the trick here is instead of taking these fluorescent probes with these wide spectra, you take isotope elements, heavy metals that don't appear in any cells, and these give you a mass peak, so you have no overlap, and then you shoot the cells one by one through a mass cytometer, and you get this cloud of ions, which you quantify for each cell, and you can get up to 45 dimensions measured simultaneously into millions of cells. This thing runs at about 500 cells a second. And here's the big thing. Instead of having this ugly spectral overlap, which we couldn't use for any quantification, we get these nice, beautiful separated mass peaks. So we can actually get quantifiable information using these antibodies, and we can finally try and do something with it, even though I will stress that this data set is still very, very, very noisy. So one of the questions we wanted to ask is, again, the T-cell system. So again, showing that we can learn the structure is a little bit boring, because particularly for the antibodies that work well, we already know the structure. But what we don't know is how this structure works. The signaling molecules, they actually have the same structure in many cell types. And if you do all these tests and say who's connected to who and who's influencing who, you'll get the same answer. And yet you give 2 T cells the same signal exactly, the same stimulus. You put them in the same tube and put them one next to each other and give them the exact same stimulus. And an effector T cell will be different than a naive T cell. A CD4 T cell will behave differently than a CD8 T cell. So even though they have the same network, even though if you draw their same network diagram, even if they have the same stick figure nodes and edges, their response is not just a little different, but a completely different decision, a completely different phenotype. So we wanted to dig in and ask, well, how does it differ? What's going on in these edges? How is these networks computing? And we felt that now that we had some quantitative information, now that we could actually trust our measurements to some degree, we can try and have a go at it. So particularly, we focused on naive and effector memory T cells. The naive T cell is a T cell that hasn't yet figured out that it needs to respond. It needs to figure out whether it's recognized something that's dangerous to the body or not, and whether whether it needs to be a guardian of the body. And it really is trying to still figure that out. An effector memory cell already knows that it's a guardian of the body. And if it finds its antigen, if something binds to it, something bad is going on, and it needs to immediately respond strong and protect the body. And so they have different questions, and therefore when they're stimulated with an antigen, when their TCR is engaged, they respond completely differently because they have a different role in our body. And so we devised a surface panel that could distinguish between these two cells, and we stimulated them at the same time in the same tube to maximize the differences. Another thing I would state is in this technology, tube-to-tube variation is so big because there's so much bias in how you poke holes into the cells and the cocktail that stains them, that it's very hard to compare two different tubes. Therefore, it was very important to do everything in the same tube so we can get a direct comparison of these two cell types. So we had the surface markers to tell us which type of T cell are we looking at, and even an effector, a CD4 or CD8, and as I said, each has a very different role in our immune system. And then looking at 11 signaling molecules, asking what are these cells doing. Now I showed you earlier how we can learn from one molecule to another, and this is real data now. And CD3 zeta is at the top of the cascade. It's one of the first things that gets activated when the TCR is engaged with antigen. And one of the things that it does is it activates SLP76. So this is real data. Each point is a cell, scatter plot of these two variables. And this doesn't look like that nice, pretty correlation. CD3 zeta very directly, very effectively, very strongly is supposed to be activating SLP76, and this pretty much shows you just how wonderfully noisy our data is. What's more disturbing is this is before stimulation versus after stimulation, and again, there's you could sort of train your eye and say, yeah, this is a little bit more peaky here. That looks a little bit better, but that's not the type of differences you'd want to see in a scatter plot between on and off. We were actually, the Nolan lab wrote this data off as garbage and an unsuccessful experiment, except it was one after another, and this was actually the best they got. So we started looking at this and thinking about it more and more and trying to understand what's going on. And first we decided, okay, a scatter plot is the wrong way to look at it. Let's look at it as a contour plot or probabilities, a density distribution. So we said, okay, this is actually looking at the density of the cells. This is the probability distribution underlying the data that we have. And what we noticed is actually most of the cells are here. Most of the cells respond this way. It creates a huge amount of density right here. And combined with the noise, and there's a lot of noise in this technology, it's not that accurate. Basically, most of our signal was coming from a single canonical point in the density of the most frequent canonical response, and the noise rather than the biology around that. And that's what was obscuring our signal. And it wasn't just in this case, it was for every pair of variables, and it wasn't just in this biological system, it was for every biological system we were looking at. So this was I'm giving you one example of where it was a pretty canonical problem. And when you look at it, it looks a little bit better than the scatter plot when you look at it in KDE. So here, this is the before and after stimulation where you couldn't see that much difference in the scatter plots. But one of the things that you could see here is that the entire molecule shift, both X and Y go up together, but you can't really see much of a difference in the relationship itself. And we decided, well, a lot of my PhD, what Bayesian networks like to do, the biggest trick that we have is that we condition on another variable. And since what we really want to know is how X influences Y, just let's condition on Y. So all I'm going to do here is I'm going to renormalize based on the conditional density of X. and see how Y's behave for each range of X. And I'm doing it in sort of a stepwise fashion. Actually, I don't do it with bins, but locally interpolate it. But when you condition on X, suddenly you see something that looks like a lot more like a sigmoid, a lot more like the type of biological response that you would want to see. The reason this is because now we're giving a lot of weight to all the huge range of variables. We're now really seeing how our target molecule, SLP76, is acting at every single range of CD3 zeta and what the function is. And we have so much data that in each one of these slices, we have hundreds of cells and enough to estimate what's going on for each region of data. And that's also the power of having so many cells. When you're working with so few cells, the rare populations, they're 1, 2, 5 samples. But when you're working with 20,000 cells, then these rare populations add up to a big enough number so that you can see, well, what happens at the full dynamic range. And now you can see that there is a very big change in signal transfer. So this is just as before, the pre-stimulation and the post-stimulation, but now renormalized or conditioned on CD3 zeta. And you see that there's a much bigger change in the actual activation or signal transfer. And that for the same increase in X, CD3 zeta, you have a much bigger response in the molecule, a much bigger response in Y. And this is in this particular case because of all the other activated cofactors that are activated in the system that we didn't measure. So we can truly say not only that X influences Y generally, but when TCR is activated, when the cell is engaged in a TCR response, the strength of this relation, the influence of X on Y is much more dramatic and much stronger. And we can actually try and quantify that using an adaptation of mutual information. So again, if you use the classic mutual information and many variations on the theme, it all gets stuck on that density, which is, again, most of the variation around that density is driven by technical noise of the technology and not by biology. If you use all sorts of techniques like adaptive partitioning and many of the other techniques to try and learn based on the density, again, they adaptively cut up all these little regions inside the noise and basically model noise very effectively. But if you do this basically on the resampled data, modeling the information of the conditional density rather than the joint density, then you can actually get real numbers for how much information you've transferred from one variable to another. So just to summarize, if you look at correlation or mutual information, you don't have much difference pre and post-stim. Kernel density estimation shows a shift of an entire population that's dominated by a single peak and noise around it. And with our approach, we can both visualize and quantify the function and how it changes under stimulation. So now we could use this directly to compare original question, the naive and effector T cells. We use it to compare other systems as well. We use it to compare diabetic T cells and healthy T cells, or CD4 and CD8. But generally, we can now really compare different types of cells. And one of the things that we see, and down here, purple signifies how much information there is or how strong the interaction is. And we see that the naive cells have a lot more information transfer, or said more sensitively, they're a lot more sensitive to how much input or there is. There's more information being transmitted from the input to the output, as you can see by the slope here. And in effector memory cells, they just go on, but there's not much sensitivity to the input. You don't have much information about how much input went into the system, how much CD zeta there were. And this continues down the entire cascade. So if you look at every pair of molecule down the main signaling pathway that transfers information from the surface into the transcription factors and down to the nucleus, you see that at every step along the pathway, the naive cells have more information transferred, which is transmitted for a longer time period, and that's makes a lot of sense given that in one case these guys have to figure out something. They have to figure out very sensibly if they're going to be autoimmune or if they're attacked or if they're recognizing a pathogen. And so they need to really be careful about what input they're getting, whereas the effector memory cells, they just need to turn on when they need to. And this is actually the opposite of what you'd see if you just look at the variables themselves. So again, because the effector memory cells have a quick turn on, their variables, if you look just at the values of the proteins themselves and not at the strength or the information on the edges, you see that effector mem are much higher because they're insensitive to the input. They just go up. Whereas the naive cells just have more information transferred along the entire cascade. We look at other nodes in the network to see what might be causing that, and we saw that in effector memory cells, they have a much stronger edge from an alternative pathway coming from PAKT, and that this is even boosted at the very beginning. They're more poised to respond because that AKT edge is sort of already on before stimulation, and therefore they're more poised to respond the moment they get any stimulation, and that stimulation gets boosted by the AKT. So then we decided, okay, one of the problems with working with biology is that it's messy and noisy and icky, and you have to take a lot of shortcuts, and you have to do a lot of things that some of my more formal computer science mentors would shrink at because it's not quite something provable. But in biology, we do have a proof that's called the validation. We can actually go and say, did we actually learn correct biology? Did we actually learn something new? And we said, okay, now we have a direct prediction. We're saying that the influence of, in this case, ERK on S6 is much stronger in naive cells than effector memory cells. And if we look at these functions, if we knock ERK down from here to here, then we expect a much bigger drop in S6 in the naive cells than what would happen if we knock ERK down the same degree in the effector memory cells. So now we have a prediction, and we can go and actually knock ERK down in a conditional knockout mouse and see if we were right. And we even have a time series so we can say when this difference can kick in. We did this in 2 replicates in 2 different mice. And what you see here is the difference between before and after knockout. In blue is the naive, in red is the effector memory cells. We were right both about the timing of when this kicks in, as well as, again, about 40-50% more effectivity in the naive versus the effector mem. And we managed to validate a similar validation for CD4 and CD8 cells and a couple other differences using the same approach. So we are able to predict differences in edge strength between these different things. So I want to tell you a little bit about the underlying method, since I know you guys are at the end of the day computer scientists. And I threw the words. We just measured the conditional probability distribution, the density estimation. I sort of threw these words very simply, as if it's simple to do it, and actually doing it correctly mattered a lot. And I, until this project, had absolutely no respect for conditional density estimation. I thought, okay, all these non-parametric things are the same thing. You throw Gaussian balls and you're done. And it's very simple and heuristic. And I read up a lot of the literature and found out that there's actually not that simple. You have a trade-off between speed and using a full kernel approach. And with the number of cells that we were doing, speed was of essence. We had 20,000 cells, 200,000 cells. That's a place where you can't sort of cut the thing. Now, the histogram approaches work really fast, But the histogram approach gave you completely different answers as you change the bandwidth, and I don't like getting different answers that are too sensitive to the input parameters. And then I encountered this really lovely paper by Botov in the Annals of Statistics doing a hybrid method where you sort of build a histogram from the initial data and use a rule of thumb to estimate the bandwidth, and then that histogram is smoothed. Now what's really nice is that he found the connection between this Gaussian kernel, between the density estimation and heat diffusion inside a bounded space. Because basically, what are you doing with these Gaussian kernels? You have data points, which could be like little spikes of heat, and you want them to diffuse. You want to take the Gaussian kernel and smooth them out. This is exactly diffusion. Now, the wonderful thing is that these physicists have been thinking about math for many more hundreds of years than we have, and they found out that, yeah, if we actually try and estimate it based on summing over all these Gaussian balls, then our computer, we need a supercomputer, but that actually can be rewritten as a Fourier series in the cosine domain. So we can actually work in the frequency domain to solve this really lickety-split. So showing you how it works, you have the frequency domain and these little binned histogram. This is the data. And you you transfer it into the frequency domain, and now you have the same data inside the frequency domain. You basically do the equivalent of you doing these Gaussians is a low-pass filter to smooth the frequency domain, removing all this noise and keeping these top frequencies, which you're hoping are the signal. And now you can take it back and invert it back into the frequency domain, into the histogram domain, and you get this nice, beautiful red smooth curve. And again, this is really basically the justification from this comes from all the stuff that the physicists have done in solving diffusion and heat domains. And now we can see an animation of the algorithm goes, over time as the thing diffuses. And basically, rather than having a width of your Gaussian, you have time. This thing eventually flattens out, so you have to stop it at some point, but that's how you sort of get the smoothing in your frequency domain. And again, it actually matters. So I remember we said this to the reviewer. We thought everything we were doing was heuristic. It was one of these proper statisticians that loves having things proved, and we had 0 proofs in our entire paper. And he said, why don't you do it in a principles fashion? You need to use Hall's method. So this was a supplementary figure from our paper showing that Hall's methods gave really bad results, not to mention took a long running time. And it took me a while to think about why the Hall's method, which has all the proofs in the world to be the most accurate method, works so poorly versus the Botov method that has no proof whatsoever. And the thing is that all these proofs assume that we can model with our computers real numbers, and that we don't round these real numbers off at any point in our 64-bit representation. And when you take beautiful, elegant mathematical proofs that assume that our computers are based on real numbers, they sometimes break down. So we decided, okay, that was fun, but in a way, let's try and take it to the next step. And the next project I'm going to talk about is something a little bit unpublished. And this was again led by Smita, the same one who developed the last one. She's now an assistant professor at Yale. She just started a few months ago. Roshan, a graduate student in my lab, and Nevin, a graduate student in Bird and Bob Miller's lab at Zurich. And the idea is we wanted to go beyond a single state and say, how does signaling rewire when a cell changes its state? In this case, the epithelial to mesenchymal transition. This is thought to model metastases in cancer. It's now understood that it's not quite the best model for metastases, but it's a great model where the cell moves from a very nice behaved, round, structured cell with very good behavior that responds very accurately to all the signals that it's giving to its environment, to an unstructured, unpolar, unsymmetric, invasive tissue that just is capable of mobility, invasion, and all sorts of really, really nasty behaviors that you wouldn't want a healthy cell to have. And it's the same cell, and you can trigger this transition by giving it TGF-beta. You can give it a signaling molecule and induce inside a plate these nice behaving epithelial-like cells to transition the very same cells and suddenly be nasty and the Dr. Jekyll, Mr. Hyde transition. And we wanted to understand how signaling changes along this transition. What causes the transition, what drives it, as well as now how are the signaling networks inside rewired so that it responds to signals and stimuli so differently. And one of the first things we realized when we are looking at it is we don't have two cell states. We have a transition. So this is time periods. Each adherent, that red thing over here, is the main marker for epithelial cells. The momentum is the main marker for mesenchymal cells, and each column here is a different day. And so the cells here are epithelial, and the cells down here are mesenchymal. But we see that there's a transition. There's a lot of intermediate steps, and particularly if we take some intermediate days like day 3 and day 4, we see that there's cells all between. And when people take time series, they actually don't take epithelial cells versus mesenchymal cells. They take different mixtures of epithelial cells, mesenchymal cells, and transitioning cells. So we wanted to take the single cell approach to study this. And we wanted to look at the entire transition. So I'm going to quickly give you an algorithm that we developed to actually get a transition where we say development, whether it's healthy development or aberrant development and change of transition, is actually a continuous approach. And rather than having a couple discrete cell states, we should actually have a continual curve, and we want to place every single cell along its place on a transition. Studies of single cell data have now shown that this sort of transitional gradual model is a much more accurate model for what's going on. Now, the challenge of actually finding this through data is the nonlinearity of the system. So many times what you have is markers go up. So if you look at even marker A and marker B in two-dimensional space, and this is data modeling B cell development, and if you have these two spots right here, these two spots right here, they are very near to each other in the scatter plot, but they're actually very far in development because first marker A went up and down before we even reached here. And you add the noise, and it's very, very hard to trace this twisting and turning nonlinear curve through high-dimensional space. And the way we decided to approach this is actually model it as a graph. So each cell is a node in the graph, and we connect graphs that are very, very similar, that express very, very, very similar levels of all the proteins. And our underlying approach here is that rather than look at the distance and look all the way directly in some Euclidic high-dimensional distance, we will take the distance as a walk along the density of our graph. And the reason is we actually believe in these small steps. We believe that if two cells are very, very similar, then they're also very, very close in their state and in their developmental path. And now by taking this walk, we're actually walking through cell density, through regions in where cell states exist and are dense. Of course, you need additional tricks to get this to work because remember I told you this data is noisy. If you're looking at a shortest path algorithm, the first thing you're going to get is shortcuts in noisy data, and any shortcut will kill you. We've since developed quite a few tricks to get rid of these shortcuts, but the first algorithm that we published that we're going to use here is basically we have two tricks or two points of randomness. In one, we're actually going to put a lot of waypoints throughout the graph because as the walk zigzags through this, it loses accuracy because it sort of zigzags through the thickness of the graph. And so by scattering a lot of waypoints and measuring the distance of every cell to every waypoint and averaging these distances, giving the nearer waypoints a higher weight into placing where the cell is, we actually can get an accurate and reliable measure of where the cell is along the entire trajectory, and we don't lose resolution as we get distance from our start point. And by actually sampling, by taking like a K and N graph, K equals 50, and sampling even 50 subsamples of that where you take a subset of those K, let's say 5 or 10 neighbors from the 50, this actually removes most of the shortcuts. So any shortcut that we might have given by one of these edges appears in only a small number of these graphs and averages out when we average over the 50 or the 100 or however many we took. And this resolves in something that gives you a lot of robustness, a lot of accuracy. I was personally surprised in how accurate and robust it was, and it allowed us to really answer and study B-cell development in a way that to a resolution that's never been seen before. So we applied this to the EMT trajectory, and this is the trajectory from blue early cells to red cells. And the other thing is we took the Botov method and did a little bit of tricks of our own. He developed it for 2D. We needed a few tricks to develop it for 3D. So here we see this sheet, sort of the density of the heat from basically P38 and PMEC and their influence on JSK3B, and you see a sort of ore-like structure here. But what was really fun about going 3D is that we could give one of those dimensions as EMT time, or time along that trajectory, and therefore we can actually see how this edge or this influence changes as it goes along EMT time, as it goes along the trajectory. And what you're going to see is that edge gets stronger and then get weaker. And again, we quantify how the dreamy and the edge strain and how the relationship changes over time. In this case, the relationship decays. Here's another case of two variables, ERK on snail, where you could see an edge forming. You can actually see an interaction happen as ERK begins activating snail in the course of this EMT trajectory. And again, this actually traces the information, or the dreaming score, the mutual information over time, and it basically gets turned on. And what's fun about this is we're taking this from a single snapshot. So this dynamics is not over a time series, but we're taking a single time set. We're taking a single time snap. We're taking data measured on day 3 and just taking the variability and the progression that each cell has had along this trajectory from epithelial to mesenchymal cells, and actually getting the dynamics of the trajectory from a population of cells, each at a different position along this dynamics, to actually infer this dynamics. This is extremely robust. So, of course, it's robust across 3 replicates. I won't show you that slide, but the timing is robust across 3 days. This is day 2, day 3, and day 4, and it has a very similar shape for each of these days. So this EMT time is sort of something that really depends on where you are along this transition rather than what day you were sampled from. And here's the sort of correlation matrix showing that really it doesn't matter what day you came from. It matters where you are along that transition, which we can infer day after day. And so how can we validate our approach? We're going to do the same thing. Dreamy represents an edge strength. So it states that as the edge strength is stronger, when we say dreamy is higher, a knockdown of that should have a bigger impact. And we can test that by drug. So again, the purple is our dreamy prediction for the strength between MEC on ERK a long time. And blue is the actual experiment of using MEC inhibition and seeing how the impact of MEC on ERK changes over EMT time. Again, a very accurate correlation. Here's another one, and we validated 5 of these. Here is 70S6 on NF-kappa-B. And one of the points I really want to make here is that in this case, there's quite a few things that influence NF-kappa-B, not just this molecule. So black is the actual level of the measured molecule. So it is uncorrelated with our prediction on how a knockdown of AKT should influence it. Knockdown of AKT traces the actual effect that we predict very, very accurately. So we could get these predictions. We can also try and say what's driving the signal. And again, in these signaling pathways, we have TGF-beta at the top, and there's a lot of pathways that can go through. Go through ERK, it can go through P38. It's actually thought to, the textbook will tell you that it goes through AKT. But according to our data, what you see actually is that a peak in dreaming and information driving through the MEC pathway. And every test we did showed that it seemed like the MEC pathway was driving this and not the AKT pathway as the literature told us. And so we actually took a long-term, this is actually after 7 days, to see how much changes. So under control without TGF-beta, there's only 2% of the cells that are mesenchymal. You give TGF-beta, you send, most of the cells are no longer epithelial, and about 50% of the cells make it to full mesenchymal ugliness, Mr. Hyde. If you actually inhibit the TGF receptor, you get something very similar to control. It doesn't matter that you gave it TGF. If you give it MEK inhibition, it's not the only signal. It doesn't stop it altogether. It's not like giving it a TGF inhibitor, but you still knock it down significantly. You get less than half transition by knocking down MEK. And when you knock down AKT, which is what the literature said should have done it, and what our data said, no, it's not doing anything. It's, if anything, increasing it, even though I think that's just variability and just, a drug perturbation. So again, we could actually have the data say that in this specific cell line, it's actually MEK. And what is the difference? Well, I don't think the people who wrote most of this literature comes from Bob Weinberg, and he's a Nobel laureate, and I'm the last person to say that he's wrong, except he did most of his experiments in a different cell line. And so what drives EMT is different in every system. And this gives us a way to say that in this system, MEK is driving EMT. And in the HMLA system, where 80% of the work on EMT has been done, it's most likely probably AKT. So what about the big picture? And I just want to spend the last few minutes telling you what I'm most excited about. One of the limiting factors here about studying AKT is that we were only looking at the signaling molecules that have known for decades to play a role, because these have the antibodies which we can measure in CyTOF. And CyTOF is not only limited to only 40 markers, but it's limited to 40 markers for which we have working antibodies, which really limits us. And we can't explore the unknown, and we can't go beyond the 35 players that were measured here. The 45 I said was as CyTOF improves. This was slightly older data. So we want to go RNA-seq. But for RNA-seq, we need many, many cells. We know, and you guys are all machine learning, we're not about to learn beautiful biology and networks out of 96 cells off a fluidine machine. So one of the technologies that we're using is based on droplet microfluidics. It was published by Klein and Mazutis in Cell 2005. Now, Mazutis is a scientist in my lab. And the idea is you have a droplet microfluidic device. There are beads coming in through here. These beads are coated with primers that have a unique cell barcode and a unique transcript barcode. Cells are coming in through here. And I'm going to animate and show. You see that cell just popping in? Sometimes you see a little white thing coming in. That's a cell coming in. And each of these cells gets encapsulated in a little microfluidic bubblet that actually does the entire code. So after they get it encapsulated and captured here, they come out of this machine as transcripts coded for their transcript molecular identity and their cell identity. And the idea behind this is that we can do about 30,000 cells in one experiment. It's it's quicker, it's more cost efficient, because these are tiny microfluidic droplets, so you don't need many of these really expensive reagents. I run a lab, so I can tell you that place is just like an incinerator for cash. One of the dirty secrets of single-cell RNA-seq data, and that is really important for you, this is the data that was published in a recent Nature paper, which, while I'm going to show a nasty figure of it, I actually think it's a beautiful, well-done paper. So I don't want this one figure to give a wrong impression of what I think of this paper. If we, again, switch, not even conditional density, let's just switch from our scatter plot visualization to our KDE representation of the data. It doesn't look that correlated. These are sort of two cells. The problem is that there's an incredible amount of dropout. So what we're doing in single-cell RNA-seq is not getting a full transcript profile of every single cell, but we're sampling between 2 to 8% of the transcripts in the cell. So rather than having this sort of matrix of cells by transcript, we have huge amount of dropouts. We have a sample of this matrix where we're sampling, let's say, about 5% of the transcripts in any given cell. So what we have is single cells and 5% of each of these single cells sampled, which is what makes things hard. So I want to finish with some magic, some imputation of a single cell data. So work again by Smita and David, a postdoc right now in my lab. And this is the same EMT data as we showed before, now using genes with RNA-seq. So if you remember that beautiful transition of EMT from E-cadherin to Vimentum, that was really beautiful. These dots are again colored by their EMT state, and you can see that this is really ugly. You don't have that beautiful transition. We're actually going to use neighboring cells, and the neighbor graph I showed you earlier to actually use information from neighboring cells and use, again, the process of diffusion to learn transfers. This is the diffusion process happening. This is a completely different algorithm than the BOTF I showed you again, but again, it uses a diffusion on this network map and diffusion of information on the network to get you the nice imputation of E-cadherin to Vimentum along the EMT. Again, we're estimating all this directly from the neighbor graph and diffusing on it. Devil's in the detail. And the diffusion map is really robust to dropout because you're looking at neighbors across so many genes at one time and so many neighbors. And that's where this diffusion really helps. And this is one case where we sort of take an original correlation matrix, we drop it out and re-infer the structure, even though almost all the correlations have disappeared. So this is the correlations of the original data, which almost disappear in the dropped out data, and this is after imputation. This is an old version of our algorithm that doesn't work nearly as well as the algorithm that we currently have. So again, if you want to see how these genes can change over EMT, this is the raw data. We don't see really beautiful trends of how these genes are changing along EMT, and this is our imputed data. And you can see the behavior, the dynamic behavior of each of these many players. And for the players where we actually know a little bit of biology, we got it right. So again, I'm sorry for telling you the magic story really quickly, but I just wanted to give you a taste of that. I thought you might enjoy. And again, thank you. This is mostly work done by Smita with Roshan and the EMT project, and David and the Magic project, and a lot of wonderful people in my lab, and thank you very much.
So we have time for questions. Surely there must be many.
Everybody.
So in the first part of your talk, you try to see how X changes Y, right?
Yeah.
And you do the conditional distribution. So did you try ordinary regression or something?
Yeah, ordinary regression worked really bad. So because of the noise, and again, so if you go, if you sort of see this structure right here, then there's a lot of noise around here. And because this is such a high density of your data, and because the noise is not necessarily completely Gaussian and pretty, many times, the regression line will not necessarily, like the endpoints here are not strong enough relative to the noise here. So the regression line can really go completely off course in these densities.
I mean, did you do nonlinear regression, say kernel regression or something, and then you can take into account the
so any method, and we tried a number of different methods. The basic problem is that most methods, most statistical methods, actually in their assumptions say, I care about where most of the data lies. I'm going to be driven by the data. Every point, no matter where it is, has uniform vote. And therefore, all these cells right here, where most of their signal is noise around one point, are going to actually drive your signal. And no matter what functional form of your model is, the idea that most of your noise is around a single point and the noise around it, most common methods that look at the joint distribution will do whatever statistics you want to do on the joint distribution. It is largely driven by a noisy density around a single area, And that part doesn't represent the full dynamic range. And that's the problem, irregardless of what statistical method you want to use. And here we're saying something that's very different from what's usually said. No, most of our data is not representative of the full span of biological diversity. If we look at the data density and give every point a uniform equal vote, we will go the wrong path. That is a pretty rebellious statement to make, but it turns out very beneficial for biology.
So I'm struck with how in a number of these experiments you have measurements at multiple time points, like you're talking about this transition. And then what you've said is, well, any one of those measurements, given the heterogeneity of the data, I can actually develop a whole time series.
So for the first system, no, because it was actually a very rapidly acting signaling pathway. And there, the time points here in the T cell thing was at 30 seconds a minute. But many of the developmental systems that we have, where things are developing over days, and cell phenotypes are supposed to change. So one, we're looking at a lot of native tissue in the body, so we have quite a few processes where our own tissues are in homeostasis. So the original B cell project was, we drew bone marrow of someone who sadly was so poor he was willing to undergo that procedure for money. It's legal, but I almost feel it should be illegal, or that these poor people should be paid more, because I know how much I paid for that bone marrow, and I certainly, and I'm sure other people made money along the way. I'm like, I almost want to pay the poor guy more if I could. But there you actually have, like in each of our bodies, we have right now all stages of development of our hematopoietic system. And actually, we've expanded this bone marrow now to get full hematopoiesis. So we've gotten a branching version of Wanderlust going, where we can go from the stem cell to the myeloid, into different myeloid fates and dendritic cells, and we just get, from a single draw of bone marrow, like a complete map of hematopoiesis and all its branches. I'm not showing this here because I had to pick and choose. For transitions that we do in a dish, again, we always see some cells don't transition, some cells part transition. Certainly for EMT and for other transitions, I've seen that we have a population. If we look at the single cell level, you can actually many times get the full dynamics. I mean, you have to choose the right day.
Yeah.
If you're too early or too late, then you don't get it. But anything in this system from day 2 to 4, we got everything.
Right. And so then in some of these systems where you see these big clouds of data, we see, well, this is really heterogeneity. Some of that in the right system, given the underlying biology, is actually kind of a map of the time course. And so, and sometimes it's not. Sometimes it's noise or other things like that, and that's an interesting thing.
So when we're looking at RNA, we're looking at everything. We actually see that there's not all the genes sort of follow this nice trajectory, and we can actually decompose the genes to genes who are involved in processes that are largely driven by this EMT transition, and to genes that are doing other things that don't relate to the EMT, but have variability of their own for how stressed the cells happen to be, or some metabolic, how much they're locally in the plate sensing. We have strong plate effects for where they are in their metabolic state. And that's actually sort of different trajectories. So one of the things we're doing now with MAGIC is actually decomposing the gene space and saying, okay, these genes which are driven by EMT should learn based on neighbors of where they are in their EMT place, and these genes who are driven by where they are on the plate and metabolic nutrient stuff should learn based on neighbors that are in that state. So we've actually taken and built a decomposed version of DREAMY that actually can work on a lot of different axes that sort of basically a composition of factors that play a role, EMT being only one of them, which was really dominant when we were focused on molecules that were based on it. And then when you look genome-wide, it's certainly the strongest signal of the data. The first principal component of the data, of the gene space, is this EMT trajectory, but there's a lot of other processes going on that are tangential.
So when you're looking at nearest neighbor and you're sort of scoring that, what are you using as a scoring measure for nearest neighbor?
So one of the reasons I didn't go into that detail was in every single system, it's completely different. For the first one here, the B cell example, we used a cosine distance because what we found is actually the proportions of variables, or the fraction of, there were big cells and little cells that were messing us up, and the cosine differences actually allow us to say the ratios between these variables. And so the cosine distance was the first deciding factor. The EMT data was really, really noisy, and it was almost impossible to get rid of all its shortcuts. So there we did another trick, and we actually used the Jacquard distance. So what we did was based on, and at this point it really doesn't matter what you use. We use cosine Euclid, it always gives you the same thing. So you take your first graph, give it 50 neighbors based on whatever distance metric you choose. And then after you build that first graph, build a second graph where the intersection of the neighbors is the edge weights. And that sort of really cleans up the data. And so basically the second graph, the EMT graph, is based on a two-step process where first we use, and it's so robust that you can use whatever you want. We used Euclid, and then the Jacquard, so based on number of shear measures, really gave it a very nice shape.
Okay. All right.
So you mentioned that your results are showing that EMT is driven by MEC pathway, but
In this particular cell.
right, and in the other systems they say that it's AT. Can you describe the difference between the cell line and?
So what these cell lines are all derived by cancers. And each cancer has its own set of mutations, and each cancer is really its own beast that has been changed and rewired from the healthy human cell that we have in us. And not only in this system, but in your response to MEK inhibition, in your response to interferon, pretty much, we've actually studied melanomas and breast cell lines where we take different cell lines that are even pooled in the same pool. These are BRAF positive P10 melanomas, and their wiring in terms of response to interferon is completely different. So these mutations have caused the basic pathways in cells to rewire in creative, wonderful ways, or horrible or satanistic ways in each individual cell line. So each cell line, due to its unique set of mutations, is its own beast. You have to learn a network for it entirely de novo. Or not entirely de novo, but there's a much more rewiring than what most cancer biologists who like to look at cell line X and say, this is the canonical case for all of human, would want to think.
No, so I understand that there are differences because of mutations. Do you have any idea why there might be these differences? Are there other differences in the cells that might explain why you have?
I think that at this point, we don't understand it enough. And the thing is that the top pathway, TGF-beta, is known to be somehow wired to both MEK, AKT, and P38. So the fact that TGF-beta can influence each of these three pathways is well known, and in different systems and different cell types influences them differently. As you saw, the strength of that relationship differs across systems, and I think that what's causing the strength of that relationship to change is additional unknown factors which we didn't measure. We have about 10,000 signaling factors in our body. Large numbers of them are completely uncharacterized, and here we were looking at about 20-something. We searched certainly within the measured variables very extensively, so the answer is not in the variables which we measured. I think what changes the strength of that initial edge is some other factor inside the cell that we didn't measure and likely don't even know what it might be.
But possibly related to TGF-beta or the signaling.
We measured most of the known molecules known to be related. I would say that it's more likely, we didn't measure everyone because not everyone has a good antibody, but it's more likely something that's not canonically associated with TGF-beta than something that is. But again, that's just my gut feeling.
Yeah, got a negligible number of the actual signaling molecules you measure. 20 out of
Exactly.
10,000.
Just probabilistically speaking, it's more likely something we didn't measure.
Yeah, right.
Okay, well, thanks again.